completed your assignments? You guys haven't uploaded the assignment, I think.
Yes, sir.
Was you also haven't uploaded the assignment? Huh? I had given questions, but you guys haven't uploaded it. Okay, anyways.
So,
Yes?
I was absent for 9 days for school, right?
Yeah, you have been absent in the school as you
Yes,
told
sir.
me. Yeah.
So I'm completing that notes of it. So I'm not writing the assignments, sir.
Okay. So that is the reason. What was going on? What
Yes,
is going
sir.
on in the school? Which chapter is going on? Revision or like what is going on?
It's motion and time graphs, sir.
That is going on.
Yes, sir.
Okay, so that is a chapter we will have to take up in the bash, I think. Motion and time, that is there for you now. Okay,
Yes, sir.
although the revision will also be continued in the bash. Okay, na, along with
Okay,
the
sir.
what is required for you.
Okay, sir.
Okay, yeah, guys, let us do these questions and then we are going to continue further. Okay, so which of the following is a strong base here?
Sir, in chat box.
In the class, you can answer in the class. Only you two are here in the class.
Sodium hydroxide.
So, Priyanka, what about you?
Yes, sir.
That is correct. In fact, sodium hydroxide. Good. Next.
Sir, acid.
Sour things contain acids in them. Good.
Sodium chloride. So third question, I'm not sure.
Not sure. It's a simple one, by the way. Sodium chloride. What is it basically? How is the sodium chloride formed? No.
Common salt. So no change in color.
No change in color. No. Okay. Can you guys provide me the chemical equation of the reaction which forms sodium chloride?
Common. Sir, acid plus base is equal to salt plus water.
I'm specifically talking about the sodium chloride. Which acid will be reacting with which base to give out the sodium chloride along with water? Yeah. We have the hydrochloric acid, that is HCl,
Yes,
plus
sir.
sodium hydroxide, sodium
Sodium hydroxide. Yes.
and AOH. Okay, and as a result we get NaCl plus water.
Cl plus H2O.
So that is the chemical equation. Okay, now, and the answer for this is no change in color. Why it will not be changing the color of the indicators?
So because it's not a base or a it's not
Acid.
a base or a acid. Yeah.
So it is a neutral substance. Okay, question number 4, guys. What's the answer to this?
Sir, neutralization reaction.
Neutralization.
Good. No, it's an important question, a really interesting question. Something you need to think of. You need to think of it for a while.
So I can
Why?
answer the second.
We can answer for the second one.
Yes, antacids.
Yeah, you, of course, you guys can answer for the second and third one, but think of the first one.
Sir,
Yeah,
is it hydrogen?
exactly. That is hydrogen. Okay, now. So when I write down the chemical formula of acid, now HCl is hydrochloric acid. Okay.
Yes.
Sulfuric acid is H2SO4. Okay. Nitric acid is HNO3. Okay. What is common in all of them?
So hydrogen.
Hydrogen, that is represented by H. So hydrogen is common in all of them. Okay, no?
Okay, sir.
Another observation you have to make one more thing. If the question were to be about base, I think we have a question on that. Okay, no. If I were to talk about what do bases have in common, So bases have OH in common. Hydroxide is in common in them. For example, sodium hydroxide, sodium hydroxide, or potassium hydroxide, or say calcium
Hydroxide.
hydroxide. Sodium hydroxide, its chemical formula is NaOH. Na is by the Latin name of sodium. Okay, now in Latin sodium is called as natrium. Okay, I explained in the previous class, I think, about why Latin was preferably be used in the medieval times. Okay,
Yes, sir.
potassium hydroxide, that is KOH, and calcium hydroxide is CaOH whole 2. What do all of them have common? OH here.
Hydrogen. I
When
mean, hydro.
OH here, and OH is called as hydroxide. So bases have hydroxide common in them. Okay, now while acids have hydrogen. Anything, this is a something which will be learning in much detail in higher classes. Okay, now so you don't need to take this thing like uh, don't get worried about this question. Okay, next questions, second and third.
So
The second
second
one antacids,
antacid,
and third one curd.
That is good. I didn't even notice. Abu Bakr, how are you? At least let me know like when you join the class. Okay. Okay, then we're going to continue with the indicators today. Okay. So here you can see some of the indicators. This is a red cabbage leaf. Okay. Then you have this turmeric, which is an experiment is being demonstrated with this. And here you have the litmus paper of red and the blue color. Talking about the indicators, so indicators, first of all, can be categorized into two criteria. The first type of indicators are called as visual indicators, where the color of the indicator will be changing. Okay, now, and that's how you will be able to determine that the given solution is acid or basic. Another type of indicators are called as olfactory indicator. There we have the example of this onion, vanilla extract, and clove oil. So they will be losing their smell. In acids, they retain their smell, but in bases, they lose their smell. So olfactory indicators are another class of indicators that will be either losing or retaining their smell in acidic or basic solution. That's how we identify a given substance as acidic or basic in nature. Okay, now that is one thing. But it just only tells us like this type of indicator will only be giving you an idea of whether the substance is acidic or basic. How acidic it is, how basic it is, that you won't get to know from that. Okay, now. So talking about indicators. So first of all, in exam questions might be asked about what are indicators. So how are we going to explain this? Indicators are basically the chemical substances that changes color when acids or bases are added in them. Right? That is what indicators are now. They are the
Yes.
they are the chemical substances that will be changing their color or will be losing their smell when acids or bases are added in them. Okay, that is one thing. Furthermore, talking about the visual indicator here, first one is the litmus paper. Litmus, as we know, it is extracted from lichens. Litmus, it is extracted from the lichens, as you guys can see here. Okay. So it is extracted from the lichens, and lichens, as you guys already be knowing, that this is a symbiotic relationship between fungi and alga. Now we extract the we take the litmus, we take the lichen, we dry it, and when we process it and we make it into strips of paper. So litmus, it's a naturally occurring purple indicator. The natural color of litmus is purple. So when it is being extracted from the lichens now, its natural color is purple. Later on, for convenience, it is dyed into red and blue color. Okay, now. So we have two colored litmus paper. One is blue and the another one is red. If you were to dip the blue litmus paper in acidic solution, we know it turns to red. Or otherwise, the red litmus paper will be turning blue in the basic solution. So this color change indicates the acidic or basic substance. Getting it now? That is the case of the litmus paper. So litmus paper, it's a coloring matter which we derive from lichens that turns red in acid solution while blue in alkaline or basic solution. So thereby this is how it is used. Okay, now that is one thing. Furthermore, this this litmus paper which we have just studied, is it a synthetic indicator or is it a natural indicator?
Sir,
It's
can
a
you repeat?
Is it a synthetic indicator or a natural indicator?
Sir, a natural indicator.
Hmm. Because we are making it out of a lichen which naturally grows. Okay. In the long list of natural indicator, another examples are of the turmeric, red cabbage leaf, grape juice even can be used. Turnip skins can be in fact used. Then beetroot, onion, tomato, cherries, curry powder, and whatnot. All these are examples of natural indicators. Okay. So plants or plant parts, they contain some useful chemicals in them that will be helpful in determining whether a substance contains acid or base in it or not. Say, for example, take some China rose or take some red petals of the China rose flower, like this one here. Okay, now the hibiscus comes in different color. This one here, this red one. Okay, now so take the petals of the China rose flower, boil it in water. Take some petals of China rose in a cup, add some warm water in that cup, and let it keep it aside for some time. Okay, then filter the solution. The filter solution will be of this color and it will be acting as an indicator. What we get to know that if I were to add acid in that indicator now, if I were to drop few acids in them, so the color changes to dark pink, right? Isn't it Abu Bakr and Priyanka? And when we add basic substance into it, the color changes to green. So this is how red China rose indicator will be working. Got this now? In fact, you can also prepare it yourself. That is one thing. Then in the list of natural indicator, another indicator is the turmeric powder. You guys might have noticed this thing. I have said this several times. Like if you were to eat some
Yes, sir.
food which had turmeric in it, right? And let's say if some food particles were to fell on your cloth, and if your mom washes that cloth, or if you yourself wash the cloth, so that stain will turn reddish in color. That stain turns reddish in color. Why does it happen? Because turmeric here acts as a acid base indicator. Say you have eaten some food which contained haldi in it, which basically turmeric was present in that food. Okay, now upon washing the stain on the cloth here, let us say there was a stain here, food stain. Okay, you guys tell me that what does washing powder contain? What is it made up of? The detergents
Sir,
that we use here. Which substance is found in the detergent? Yes, Priyanka.
Base, right?
sir, acids.
What are you saying?
Sir,
Acid?
acids.
No, it's
Base.
base actually. No, bases are present there. Okay, now if you guys can recall, while we were learning about the use of base, it was one of the use of the base which we had learned. Right over here. See, sodium bicarbonate. It is used in preparation of detergents. That is
Yes.
Na2CO3. So sodium bicarbonate, it's a base that is used in the manufacturing of the detergent. So which is basic in nature. So what happens upon applying the detergents on this food stain, and that food stain contains turmeric in it? What do we get to see? That the color changes to reddish. It changes to reddish. Okay, now why does it happen? Because when we have a turmeric solution and we add some base into it now, the color from yellow will turn to red. Okay, now in fact you can see here. Here we had this turmeric solution, we added some base in it, the color changed to this red. That is how it works. Okay, now. But what about the basic solution? If I were to have a turmeric solution and in that if I were to add some acid, so you guys tell me, will the color change to red or some other color or will it retain?
So it will change to yellow, sir.
Basically remains the same now. Yes, Abu Bakr, Gauss and Priyanka.
Yes, sir. Yes, sir.
So it remains yellow basically. Good. Okay, answer the following question here. Yeah. Answer the following question. What effect does turmeric have when mixed with a basic solution? That is correct. Was I'm waiting for others to answer. Priyanka and Abu Bakr, why you guys are taking this much of time?
Sir,
Yeah.
we should answer it in chat box.
Yeah, however you like, whether it be in the chat box or in the class.
Okay, sir.
Yes, Abu Bakr, that is correct. Okay, meanwhile, as you guys have answered this, answer this thing to me also. Do tell me what will be the color of the methyl orange solution in acidic substance. If I were to add few drops of acid in methyl orange, what will its sorry, not in
Sir,
methyl orange. I mean to say this one, um, China rose petals. Okay, now what will happen?
sir, in acid?
Yeah,
Sir,
if
so I think purple.
think
Dark
purple,
pink.
No, sir, green.
green, no.
Sir, in acid or base?
My question is that what will happen if I were to drop some acids into red china petal solution?
Sir, green. It'll turn green.
Green.
Sir, in base it's green, right?
In base it is green, no, actually, Gauss.
In
When you,
acid it's dark pink.
in acid it's dark pink, or you can say magenta color. Okay, now Gauss getting it.
Yes, sir. Yes.
Just let me go back to this slide once again. Here, see, in acid the color changes to dark pink or magenta color, while in base it turns to greenish. Okay.
Okay, sir. Okay, sir.
Okay. So in the list we had mainly a few types of natural indicators. We did learn about the litmus, and then we did learn about this what do we say, this turmeric powder. Apart from that, we have these olfactory indicators. In fact, these are also the natural indicators. Okay, now let's move on to the synthetic indicators. So synthetic indicators are those indicators in which chemicals will be used. They will be made up of different type of chemicals. In the list of synthetic indicators, some of the common ones are phenolphthalein, methyl orange. Okay, now. So phenolphthalein is an acid-base indicator, which is its natural color is colorless. Meaning that it doesn't have any color. It looks like water. But when you will be adding base into it now, it will turn into pink. Okay, now. But in acidic solution, it remains colorless. Getting it? So phenolphthalein is an acid-base indicator which is colorless in acidic solution. Let us say you have taken this. If you were to drop some acids in it, it remains colorless. These are drops of acids, let us say. It remains colorless. But if you were to add few drops of bases in it, thereby the color will turn from pink to red. But turns pink to red actually. Getting it, guys? The color changes to pink to red. Got this? So this is
Yes, sir.
how the phenolphthalein indicator will be working. Next, we have this indicator. Next, we have the methyl orange indicator. Okay, now. So methyl orange is a popular pH indicator that is used in the titration experiment also, like in the chemistry lab. And later on, you will get to know about the titration experiment in chemistry labs. But anyways, talking about its use, the color of the solution of the color of the methyl orange solution, it will turn red when we'll be adding acid in it. So from orange it turns to red. From orange it turns to red upon addition of acid in it. But upon addition of base, what happens? From orange it will be turning to yellow. Got this, everyone? So this is how the methyl orange works. Okay. So it will turn red when you add acid in it. Okay. Or it will turn yellow when you add base in it. So this color change signifies the presence of acid or base in the given substance. So this much should be clear. Okay. Okay, do tell me this one, like how the phenolphthalein works. What color change will you observe?
Sir,
Yeah.
Sir, sir, if we put acid in that solution, it will remain colorless, and it will, and if you
in
put a base in
base,
it, it will turn into red, I think.
pink.
Yeah,
Hmm.
yeah.
Pink. Pink. Okay, now pink, or like if the acid is very strong. If the acid is very strong, it will convert into orange red color. Okay, now let us say you have a acid that is having a pH level of like 0 or 1. So then the color will be turning to orange red. Okay, now it will be turning to orange red if the pH level is around about 0 or 1. Or if the pH level is, let us say, about let us 4 to 7, then it will be colorless basically. Okay, now but nonetheless we basically say that it will be colorless in the acidic solution. Okay, now no need to go into that part of the discussion. Just know that in acidic solution it remains colorless, but in the base it turns to red color. Okay. Then we have the case of olfactory indicators. So onion, it retains the smell if you were to add some acidic on. You can try this. You can just take a onion extract, take onion juice, and add some lemon juice in it. So thereby you will not see you will see that the smell will remain same. But if you were to add some baking soda in it now, it will be losing its smell. Then you have the vanilla extract and clove oil. They will also be working in the similar manner. Okay, now vanilla extract is very famous and it has very many uses. You guys know about this vanilla extract?
Yes, sir.
Hmm? It is used in desserts. It is used in making many dairy products. Yeah.
Sir,
In ice cream. Yes, Sprinkle.
sir, is it vanilla? Um,
Yes. Yeah, I'm listening. Your vanilla extract, it has several benefits and it is used as a flavoring agent in different kind of food and beverages. It is in fact used in baking the cakes as well, in cookies, and in fact in ice creams also. You might have eaten the vanilla flavored ice cream, right? A very common flavor in the ice cream. That is one thing. Okay. Talking about a very special type of acid. So we have the example of aqua regia, which can even dissolve metals like gold in it. Normally, what we know that acids, usually even if we have strong acids, it will not dissolve some unreactive metals. But aqua regia is a very strong acid. In fact, it is made up of two type of acids. It is made by mixing hydrochloric acid and nitric acid. Aqua regia, it's a mixture of two acids. Right now, we are learning about a special topic here, aqua regia. Okay, now we have finished the chapter, and at the end of the chapter, we are learning about a special type of mixture. Okay, now that is aqua regia. Getting it? Aqua regia, by the way, it is also called as royal water. Okay, it is also called as royal water. So aqua regia, it is made up of mixing hydrochloric acid plus nitric acid. So we mix nitric acid with hydrochloric acid. So we get a very strong solution, which can dissolve metals like gold in it. It can dissolve metals like gold or platinum in it very easily. Okay, now so it will dissolve in it. Got this now. Normally, the melting point of gold, platinum, it is very high. Normally, the melting point is very high. You have to heat the gold to more than 1000 degrees Celsius in order to melt it. In order to melt platinum, you have to heat it to more than 1700 degrees Celsius. But here, by just dissolving it in this liquid, it will melt. Okay, now. Okay, few more questions are there. We are going to complete this, and then we're going to take up the next chapter or the topic in the class. Okay. By the way, Abu Bakr, if there's any new update from your school, please update me. Have you been updated with your syllabus or any other thing?
So I have sent you in the chat box.
You have? Let me see. Okay, your exam is tomorrow. Okay, okay. So you have questions from these two chapters. Okay, okay.
Sir, these two chapters is going to come.
Okay. So it's a unit test now. I think it's should be a unit or half yearly test. It's a half yearly test.
Sir, I don't know.
Okay, okay. Anyways. Yeah, you guys start answering it in the class only. Answer it in the class only.
So first one, indicator.
Others also saying the same or different?
Sir,
Both A and B.
Saying both A and B. What about you,
Yes,
Priyanka?
both A and B. Both A and B.
It is appropriate one is both A and B. That is option number D. Okay.
So
The color of red litmus paper in acidic solution.
sir, is it C?
Option number C you are saying. Okay. What about others?
yes, sir. Option number B.
Option number B you are saying.
Yes, sir.
Abu Bakr you are saying
C.
C. So Abu you are saying C. Priyanka you are saying
C.
C. Okay. It's wrong. Actually,
Sir, is
option
it D?
number obviously it is going to be D then. I'm going to say out of A and D, the D one is appropriate here. See, the thing here is that the color of the red litmus paper in acidic solution, what happens there? So you have an acidic solution. In this, I have dipped a red litmus paper. What we know that acids converts the color of blue litmus paper to red now. So red litmus paper is going to remain same of the same color. Okay. So the answer for the first one is red. Getting it, guys? The color of red litmus paper in acidic solution is not going to change. It will remain same. That is red. Clear to everyone?
Yes, sir.
Now say
Yes.
that you have taken a neutral solution, like you have taken a salt solution. In the salt solution, you have dipped the same this one. And what do we know? Neutral substances do not change the color of any indicator. So again, it is going to remain same. That is red now. Getting it, guys?
Yes, sir.
Okay. While in the basis turns the color of red litmus paper to blue. So it's D then. Getting it? Okay. You guys got confused in this word of colorless and red. Okay, no, but the more appropriate one is red here. Okay. Okay. Others you guys have answered or not? Okay. Question this one you won't be answering. This one you have to answer. Which of the following is true?
So first one, option number D.
Option number D.
D, sir.
Okay, no.
So
Good.
second one, option number C.
Yes, sir, C.
Yes, Abu
And
Bakr?
third one, option number B.
You getting the same answer or different, Abu Bakr and Priyanka?
Sir, the third one is
Ant bite contains an acid, and the acid is
Formic acid.
formic acid. And baking soda is a base or a neutral substance? It is a
Base.
base now.
base.
So base will be reacting with this formic acid to neutralize its effect, and you get the relief from it. Okay. Okay, we have the anxiety questions also, but as you guys have updated me, like Priyanka, in your school, the chapter motion and time is going on. Okay.
Yes, sir.
Kindly others also check if you guys have the chapter motion and time in your syllabus. No,
Sir, I have it, sir. I
And
have it.
sir,
I don't have.
you don't have. Priyanka and Abu Ghaus, you guys have.
Yes, sir.
sir,
Yeah, I'm listening.
in physics, motion and time,
Mm-hmm.
and in bio, it's respiration and organism.
Motion and time in bio, it's a respiration and in organism.
Yes, sir.
Okay, so respiration in organism that has been done. Motion and time is left for you to be done. Okay.
Yes, sir.
Okay. You still haven't been updated with the syllabus, I think. Yeah.
Yes,
Think.
sir.
Okay. Anyways, that's a sad thing about the school that they don't update about the syllabus on time. Well, so what's the update on your side? You have been updated about the syllabus
Yes, sir. Sir, I gave you the syllabus in the website, sir.
in the website.
Yes, sir. The Learnivio website, sir, where you submit the assignments. I gave you the syllabus there.
Okay, on your on the sheet you have uploaded it. On the assignment sheet you have mentioned your syllabus.
No, sir. So there's like so there's like a section of
Okay,
like a
understood.
syllabus.
I understood.
School syllabus I updated over there, and I wrote the exam.
Okay. By the way, when's your exam date? At least tell me that. When is your exam?
So 29th.
29th, no? Okay.
Yes.
Okay. Since there are many chapters which you haven't studied from me, if you feel for any particular chapter that you'd like to revisit, like as you said, like you'd like to discuss this chapter acid bases also, we did discuss this. If there's any other chapter which you'd like to discuss, do let me know. And Abu Bakr, you also let me know. Abu Bakr, by the way, tomorrow you're having your exam now. So nutrition in animal, it is a chapter that is common for everyone, right? So how
Yes,
about
sir.
Yes.
we do some questions from the chapter in nutrition in animals?
Yes, sir. Yes, sir.
Okay, na?
Sir, what if we learn the chapter respiration in animals?
Respiration
In
in,
our,
okay. Today, in today's
Sir,
class, or
sir, in my school, we are doing respiration animals, sir. We're like doing the reading, but not do question answers yet.
okay, respiration in an organism is going on in your school as well.
Yes.
Okay. Are you having difficulty in that chapter in the school?
No, sir. Till now, no difficulties.
No difficulty. Okay. Yeah, Abu Bakr, what were you saying?
sir will revise the chapter respiration and organism.
Okay, okay. I was thinking since tomorrow you're having your exam, so how about we discuss questions from nutrition in animals that might help you score better in your exam? Or if you guys say we can do the chapter respiration, I'm leaving it up to you guys. Yes,
Sir, we'll do questions from all the chapters.
from all the chapters.
Yes, sir.
Okay, okay. Okay, let's do that. Okay. And Priyanka, most probably in the next class, I will be taking up that chapter. Okay, na?
Yes, sir. It's
Motion and
okay. Anytime is okay, sir.
yeah. And I think in the previous class, we were also doing some questions for this batch. Just give me a minute and then we're going to continue. Okay. Yes, you guys were there while we were doing this question answer practice in the previous class.
No, sir.
No, sir.
You guys were not there. Okay, it's from different batch. I'll be resharing my screen in a minute. Okay.
Okay, sir.
Okay, sir.
By the way, you guys define to me what do you guys understand by respiration? What is respiration? Yes. What is respiration? Why is it important? What are the types of respiration?
Sir, there is
Mhm.
internal respiration and external respiration.
That doesn't clarify it properly.
Sir, respiration is like a breaking, like the
Mhm.
Breaking food.
yeah, like breaking complex, like
Breaking up,
process.
breaking up of the food in the presence of oxygen.
Yes, sir.
Okay, now to get energy out of it. Along
Sir.
with energy, water and carbon dioxide is also given off, and some amount of heat might also be given off. Okay, that is the thing with the respiration. And we know that organisms are found to be living even in the most strange place on earth. Like some bacteria can be found to be living in the vent of the volcano. Some bacteria will be found living in the depths of the oceans where availability of oxygen will be very less. So there are some organisms that will be able to carry out this process of respiration even in lack or even in absence of oxygen. Right now, so there will be two type of respiration. Namely, they are yes. What are the two types of respiration, guys? I'm just taking some questions and then we're going to continue. Okay.
Sir, I told you that we're still doing reading, so I don't know about that question, sir.
Okay. Okay. What about others? Those of you who have already studied this, please let me know. Guys, didn't we learn about this?
Sir, internal and external respiration.
In, see, on the basis of whether they will be utilizing oxygen or not. So it's aerobic and anaerobic respiration. Yeah, that's what I'm asking now. Didn't we learn about all this, guys?
Yes, sir.
Mm-hmm. So we did learn about all these things in the class now. Okay.
Sir, aerobic respiration, it breaks down glucose, right?
Both of them, in both the places, breakdown of glucose will take place. In aerobic respiration, the breakdown of the food, namely glucose, will take place in the presence of oxygen, in the
Okay,
availability of
sir.
oxygen. But in the case of anaerobic respiration, breakdown of food takes place in absence or lack of oxygen. Okay. Okay, so we have some questions here from the first chapter, nutrition in plants. What is the process by which plant makes their own food? It's simple.
Photosynthesis.
Okay, others also answer it. That is obviously photosynthesis. Okay. Transpiration will take place via which part of the plant?
Sir, what do you call that?
Transpiration.
Sir,
Yep.
Stomata.
Okay. What about you, Priyanka? Priyanka. Yes. Abu Bakr.
Sir, the
Same.
leaf.
Leaves and leaves. Where exactly in the leaves? So in the leaves we have, as Gauss said, stomata. Exactly.
Stomata.
And stomatas have stomatal pores. Stomatas have stomatal pores in them now. Stomata are basically tiny pores in the plants. Like if you can see, like this will be the somewhat. This is how this stomata will be looking like, right?
Yes, sir.
These bean-shaped structure are called as guard cells, right?
God, sir,
These,
yeah.
these are the guard cells. Okay, guard cells. And here, this is the pore basically. Okay, and this is a basically opening, tiny opening in it. Okay, now, and this opening is called as, this pore is called as stomatal pore. The one that I've highlighted using the yellow color, that is stomatal pore. Okay, so many things will be exchanged via this pore. What are those things that will go in or come out via this stomatal pore? Exchange will happen.
Sir, exchange of gases.
Exchange of gases. So which gas will go in, which gas will go out?
Sir, carbon dioxide go in, oxygen come out.
Okay,
Yes,
CO,
sir.
CO2
Yes,
goes
sir.
in, good, and O2 comes out. Okay, in the process of transpiration, what will go out?
Sir, water vapor, excess water vapor.
Exactly. Okay, now water vapor will go out. Okay, now how does transpiration help the plant?
Sir, sir, the plant only absorbs like 5% of the water, so so like the other 95% it has to like remove it.
It will get rid of the extra water
Yep.
via transpiration. Okay, there's one more important role played by the transpiration in plants. Yes, there's one more important role played by it, which is very important. Think of it. Yes, one thing is that just like sweating cools down your body's temperature. Sweating
Yes,
helps
sir.
you to cool down the body temperature. So in the hot
Yes,
summer,
sir.
transpiration will be cooling down the temperature. So transpiration is a major process performed by the plants to keep it cool. Just because we know evaporation cools up the body. When sweat evaporates from the skin, we feel cool. That is one thing. Furthermore, it will allow the movement of the water and the nutrients from the root to the shoot of the plant. What is happening, if you guys can recall, we did learn about this in detail. For those of you who have learned that as the evaporation will be taking place from the leaves now, so this will create a suction pull. Getting it, guys? As the water will be lost in the form of water vapor from this stomatal pore, so it creates an upward pull called as suction pull, because of which water will be sucked out from the roots via the stem to the shoot system of the plant, to the leaves and to other parts of the plant. Okay, now, so it helps the plants to take up water and nutrient from the soil. So transpiration moves the water and nutrients from the roots to the shoot of a plant. That is one important function performed by it. Okay, now, so it is helping the plant to take up the mineral from the soil, because by creating a suction pull. Okay, that is an important function performed by it. Also, transpiration is playing an important part in the water cycle also. Now, if the transpiration will stop, water cycle will not happen. The water cycle will be disturbed. Getting it,
Yes, sir.
guys? Okay, moving to next question. Plants obtain all the nutrients from the soil. Is it true or false?
Sir, true.
What about others? Do plants obtain the nutrients only from soil?
No, sir.
No. Okay, no. They can in fact obtain it from other sources also. Yes.
Sir, like sunlight, oxygen.
Sunlight itself is not a source of it's not a type of nutrient, but it's a source of energy. The chlorophyll will be absorbing the sun's energy, okay, na, to help to carry out the process of photosynthesis. Plants to get nutrients from the air also, no. Plants get nutrient from the air also. We can elaborate on this. What do I mean to say?
Yes, sir.
Like oxygen, I mean to say carbon dioxide. Plants are absorbing carbon dioxide from the tiny pores in their leaves called stomata, right? Now, so they do take nutrient from the, although they are taking nutrient from the soil, but but they're taking nutrient from the air also. Okay,
Okay,
so
sir.
so that is the thing. In total, plants needs about 16 to 18 different chemical elements that are important for them to grow. Okay, next question.
Chlorophyll.
Yes,
That
sir.
is
Chlorophyll.
chlorophyll. Very good. Next.
Sir, because uh,
Yes, you guys think of this.
sir, because leaves are the one who absorb sunlight, and the uh,
Yes, sir.
and the stomata in them does the gaseous exchange and all that.
Exactly. Okay, now. Okay, here I am drawing a leaf here. Okay, and I see that this is a colorful leaf here. Okay, now there are different color patches on it. Now you guys tell me these places, this part of the leaf which is colored, which is having a different color, will photosynthesis happen there?
No, sir,
No.
because chlorophyll is not present in there.
Yes, sir.
That's not the case. Chlorophyll is present in colorful leaves also, but the thing is that they are shaded due to another pigments. Chlorophyll is present in the colorful leaves. Okay, now getting it what I'm trying to say.
Yes,
The
sir.
thing is that there are some other pigments. We only study about chlorophyll, which is the green pigment, huh? And
Yes,
it is
sir.
present in all the leaves. In fact, it is present even in those leaves that are not green. The thing is that say that we have a leaf of different colors. Say that you have a yellow leaf. Yellow a leaf has a yellow color because of presence of yellow pigment called as carotenoids in them. Okay, now or a leaf will be red in color because of the presence of a pigment called as anthocyanins in it. So the thing is that let's say that this is anthocyanin. Okay, now I mean to say this is, let us say this is the layer of chlorophyll. Chlorophyll is there, but above chlorophyll, let us say anthocyanin is present, which is the red pigment. So chlorophyll is there, but it has been shaded. It is being shaded due to the layer of another pigment on it. Try to understand what I'm saying. You guys getting it? What I'm saying?
Yes, sir.
Okay, now. So the green chlorophyll is present there, but it is being shaded. It is being covered with another type of pigment. That's why. Otherwise, you think will this plant be able to survive? Will it be able to perform photosynthesis? How is it going to stay alive? Okay. Okay then.
Okay, sir.
Question from nutrition in animals. Few basic questions are there, and then we are going to take up some more questions. Yeah.
Okay, sir. Sir, sir, in our book, the first question was not even there.
Yeah, it's basically kind of a logical question. Just think of it. So the
Sir,
following is
dual
not a
system, sir. Dual digestive system.
dual digestive system. What about others? While you think of this question, keep this in mind. What are the types of digestion we have learned?
Or, sir, incomplete digestive system.
Yes, sir. B.
Yes, sir.
Thinker? Very good. Because we have example of dual digestive system. Okay, now we have the example of dual digestive system in the case of some animals, like in the case of the ruminants, right now.
Yes, sir.
Getting it? Okay, now. Also, dual digestive system basically refers to such a type of system now. Like basically, one system will be responsible for releasing the solid waste, another one will be responsible for releasing the liquid waste via the different openings in the body. Like in the like for example, in the case of the dual digestive system or two-way digestive system, so thereby there are different openings in the body. Like we take in food via the mouth, the solid waste is removed by the anus, and the liquid waste is removed by the urethra by the penis. Okay, so food and the waste products are entering and exiting through the different point. I mean to say in the case of the dual digestive system. See in the case of most of the animals, food is entering through the mouth and exiting through the another opening here. Furthermore, in the case of ruminants also, the dual digestive system is observed there. Okay, now one where solid waste is removed, another where is the liquid waste is removed. So that is there. Complete digestive system. Yes, our digestive system is said to be a complete digestive system because every usable component present in the food is being completely digested. Okay. If you were to talk about digestive system, which is the site of complete digestion in human body?
So can you repeat the question?
The site of the site site the site of complete digestion of food in the human body.
Site.
Site basically place, area, but refers
Yeah.
to
Sir, the intestines.
which one?
So small
About,
intestine.
okay, what about others?
Sir, or the stomach.
Not a stomach. Priyanka, Abu Bakr, what happened to you guys? It's the small intestine. Okay, now the small intestine is the site of complete digestion in the human body. Okay, now let us see what happens to the food when it goes into the small intestine. So basically, the small intestine receives secretion from pancreas and from the liver, as we know.
Yes, sir.
Okay, pancreas will be secreting pancreatic juice, liver secretes bile juice. Say that this is the small intestine. So this small intestine, firstly, let me tell you what happens to the food. Our food has different component in it, right? Our food has different component in it. Fats is present, glucose is present, protein is present, etc. And all of them is finally broken down to the simplest molecule in the small intestine. So liver, which is the in fact largest gland in the body, it secretes the bile juice. Where is bile juice stored, Priyanka?
Sir,
Where is it stored in the body? Yes. Mm-hmm. Okay, anyone else?
Sir, liver.
sir, yes, sir.
Liver. Bile juice
No,
is
sir.
secreted by the liver and is being stored by the liver only. No, it's actually the yeah. I have given a hint. What is it? Oh, Priyanka, Abu Bakar, you guys are not revising. I'm sure you guys haven't been revising the previous chapters. Gallbladder.
Oh, sir.
It is important to revise the chapters which has been already done. Okay, now? Abu Bakar, tomorrow you have your exam from this chapter. Make sure to revise everything, okay? Now, what am I
Yes, sir.
saying? That bile juice is being secreted by the liver for a temporary period of time. It is stored by the gallbladder, but it basically goes into the small intestine. When it goes into the small intestine, the food that is coming from the mouth or from the stomach here. So, if any fat is present in it, the fat will be converted into fatty acids and glycerol. Okay, that is something which happens here. Then we have pancreatic juice also. Pancreatic juice is secreted by the pancreas. Okay, na? Pancreatic juice is secreted by the pancreas, and the pancreas will be breaking down the protein present in it. Okay, na? So thereby, protein will be converted into amino acids. Okay, now protein is converted into amino acids by the action of the enzymes present in the pancreatic juice. Got this? What I'm trying to say.
Sir, where is insulin present in your body?
Insulin is produced by the isolates of Langerhans cells in the liver. Liver produces insulin hormone. Okay, now
Okay, sir.
insulin is a hormone and its function is to regulate the sugar level in the blood. Okay, getting it now? Sorry, not the. Did I say the liver or pancreas?
So you said liver, sir. I think it's pancreas, sir.
It's pancreas. It's pancreas. Okay, now I thought I would say it, but I said something else. Okay, now. Anyways, so thereby the insulin function is to regulate the blood sugar level. Okay. Talking about protein, so protein will be converted into the simpler molecules called as amino acids. Got this one? So these components of food are broken down into the small intestine, and the food is said to be completely digested. Furthermore, we have carbohydrate also. So carbohydrate present in the food will be converted into glucose. Okay, now walls of the small intestine also secretes juice, and they are called as intestinal juices. And intestinal juices will be doing this work of converting carbohydrate into glucose. Okay, few questions are left in it. Please complete this, and then we are going to end the class. Yeah. Guys, few questions are left. Kindly complete this quickly. Each one of you take one question. Priyanka, you take question number 2, was third, and Abu Bakar fourth one. I suppose I'm still audible to you guys in the class.
Yes, sir.
Yeah. Question number 2,
Sir,
is it true or
second
false?
one is false.
It's false? No. Second one is true, no. Herbivores have a special teeth for chewing the plant material.
So small intestine,
No, Gauss, it's not his stomach. The breakdown of the food starts from the mouth only. The starch is partially digested in the mouth. Okay.
Yes,
so
sir.
it is to complete the entire, uh,
Describe the role of the small intestine in digestive system. Yes.
sir, it is to,
You can take the help from this, like in the screen. Yeah.
so it is to complete the digestion.
Basically, you have to be very specific in answering this. We can't just say that the complete digestion just takes place. We have to explain that the bile juice will be breaking down the fat into fatty acids and cholesterol. Pancreatic juice will convert the protein into amino acids, while intestinal juice secreted by the walls of a small intestine will convert carbohydrate into glucose. Okay.
Yes, sir.
Okay, work on this and revise it properly. That's it for today. We'll be continuing from next class. Okay.
Yes, sir. Bye, sir.
Yes, bye. Uh, yes, Aleph, you there in the class?
I love this.
Yes, sir.
All right. So we are going to discuss your test. You had given the chapter test of microorganism, friends and foe, and then friction, right? Okay.
Yes, sir.
Just let me take your sheet and then we are going to continue. Okay. By the way, you tell me this thing. In the chapter, are you facing some difficulty in those two chapters? In microorganisms, friends and foe? Yes.
No, sir.
Are all your concepts clear from those chapters?
Yes, sir. Sir, I had doubts in some like natural phenomena.
In some natural phenomena, there you are having doubt.
Yes.
Okay. In one of the doubt class, we can discuss that topic. Do let me know in which topics you are having doubt.
Sir, like the earthquake part. How earthquake occurs.
How earthquake occurs? Okay, okay. Basically, the one-line answer to the question would be that we know that our crust is divided into different tectonic plates. Our crust is made up of tectonic plates, and these tectonic plates are not joined. It's not a joint. They are not joined together. They are fragmented into different pieces. Okay, no, and they are kind of floating above the mantle. So there comes a time that when two plates will be meeting each other, will be colliding like this. I'll be moving past one another like this, because of which there will be a movement in the crust, and as a result of that, we get to see that this loss of life and this damages building would be getting destroyed. Okay, we'll be taking up that in more detail in the class. Okay, now right now, let me just quickly take your question paper. Okay.
Okay.
So well, if you gave the test of chapter microorganisms, friends and foe on 26th of August, right? And there you haven't performed well in that test. Okay. The thing is that you have got about 13 and a half marks out of 30. In the chapter, you have got 13 and a half, 13 marks out of 20. Okay. You see, there in question number 8, you had done mistake. Both the options A and B were correct. In question number 26, you had to be specific there. You had to mention the name of the mosquito. It was female Anopheles mosquitoes, and that causes malaria, right?
Yes.
Okay, then have you opened the sheet in your page? Do you have the sheet with you? If you have, kindly open it. There you can see the mistakes. Okay.
Okay, sir.
Mhm. Then in question number
So let me
36.
check. Do I have that test paper?
Okay. In question number 6, you were in question number 36, you were required. There you will required to explain the role of. You had to explain about harmful and the bad bacteria in question number 36. Okay. The question was you had to write short note on useful microorganism or either on harmful microorganism. But what you have done, you have just given the examples of useful microorganism as rhizobium, lactobacillus, and yeast. But that is not the right way to answer such questions. You can't just write go on to write the examples. You have to explain it now. Getting it?
Yes, sir.
Hmm. Then again, in some other questions, like in question number 37. Look, question number 37 was you had to explain the use of bacteria, fungi, and algae. So you have just written bacteria helps in turning milk into curd. Or the sentence formation is also not proper. You have written bacteria helps of turning milk into curd. So there's a grammar error in it. Instead of, you have to write into. So bacteria helps. I mean to say of, it's in. So bacteria helps in turning milk into curd. Fungi helps to give us protein. That is not the correct way to answer this question. Algae gives us supplements, biofuels. Okay, that one was correct. But you should have added more answers to it, more points in it, because this was a 5 marks question. Okay, I left a new camera on in the class. This was a 5 mark question, so there it was important to add at least 6 to. It should be at least 7 to 8 line answer. Okay, now. So this is
Okay, sir.
okay. Now also make sure to take this test seriously. Okay, now take the test seriously. Then you had one more chapter. Microorganism, friends and foe was there. Then you had the chapter. Okay, this was only there. Okay, now. So you haven't performed well in this. You need to work on the areas where you are missing. Okay,
Okay, sir.
got this now. Okay. And in amoeba, you had drawn the structure of the amoeba, but you haven't mentioned anything on that. Which part is a cell membrane, which is nucleus, and which is food vacuole. Okay. So in the subjective questions, try to add some more points. In the subjective questions,
Okay.
try to add some more points. In the MCQ questions, you have done well. But when it comes to subjective question, you need to work on it. See, as I always say that you need to work on concept understanding plus on the writing skill. Okay, now so work on understanding the concept and practice writing the answers of questions. Okay.